Welcome. Thank you for joining us today for our webinar to present you our Multiplexed platform that combines high-level multiplexing with spatial information on single tissue slides. So why does the combination of multiplexing and spatial information matter? In the context of immuno-oncology, pathologists are realizing the growing complexity they can tap into when analyzing the tumor microenvironment. Its biology contains a wide variety of heterogeneous population of cells, such as immune cells and tumor cells. The ability to capture how they interplay and interconnect within the tumor section may better inform prognosis and patient stratification. This growing need for a high number of markers is especially increased by the development of combination therapies. As an example of the value that can be derived by combining multiplexing with spatial information, here's an excerpt from a publication recently made by Dr. Bernie Fox and Dr. Zipei Feng. First, they looked at the presence of PD-L1 and noticed that in and of itself it didn't hold any prognostic power. On the other hand, when they looked at the proximity of PD-L1 positive tumor cells next to CD8 T cells, they were able to stratify patients. Those patients that showed a lower number of these PD-L1 tumor cells next to cytotoxic T cells showed a better prognosis. Ultivue is uniquely positioned on the biomarker discovery value chain. We sit on the sweet spot of this combination of high-level multiplexing from the single to double-digit number of markers and spatial resolution. Our unique selling point resides in the offering of a reagent platform that will be compatible with the wide range of instrumentation that is currently there on the market, ranging from autostainers to imaging scanners. Our reagents are meant to be used both in an upstream exploratory use case where the need for multiplexing is high and the number of samples to be run on a given assay is lower, and downstream in the routine use cases running clinical trials with a signature of biomarkers already narrowed down, so by design a lower level of multiplexing and a higher level of samples to run with. Our business model at its core relies on the commercialization of these high quality reagents. There are three ways we interact with our customers. For the upcoming year, in 2018, we will launch a portfolio of kits that will fully be compatible with the wide range of instrumentation, autostainers, and multispectral imaging scanners. These kits will be designed in such a way that they will seamlessly integrate with these existing workflows. As of today, we're engaging with customers through fee-for-services, where we receive samples that we run on behalf of our customer, either on standard panels that have already been validated internally or custom panels based on specific requests made by our customers. In some selected few cases, we also extend the service in a further partnership where we can explore further ways to deeply connect with our customer base. In a nutshell, our value proposition touches on high multiplexing in situ, the ability to record an unlimited number of markers on single tissue slides while keeping the spatial information intact. And we do so while fully preserving the antigenicity and integrity of the samples. Our reagent platform will be widely applicable to the full range of instrumentation, autostainers, imaging scanners, and is as of today applicable to any FFPE tissue slides. Our technology is also amenable to very high throughput by virtue of the fact that we rely, as you will see, on a single staining step and single amplification step, regardless of the number of markers you would be looking at on these single tissue slides. And of course, the kits that we will launch in 2018 will be meant to be fully plug and play, easy to use, seamlessly integrated in existing workflows and instruments. Without further ado, let's dive into the data that we are able to generate with our in situplex reagents. The images that you're looking at were captured from a tonsil tissue slide looking at a 5-plex panel composed of DAPI, CD3, Ki67, PD1, and cytokeratin. The image on the left-hand side is a 20x magnification zoomed out portion of that tonsil tissue. The image on the right-hand side is a zoomed in section of one of the lymph nodes of the tonsil. Going into the nuts and bolts of our in situplex platform, our technology relies on DNA-mediated fluorescence. We introduce an intermediate bridge composed of two complementary DNA strands between the antibody and the label. One strand, the barcode, is covalently attached to the Fc domain of the antibody, its complementary form, the label, to a fluorophore. Our technology's robustness is guaranteed by three building blocks: DNA conjugation that ties into the specificity of the platform, where we guarantee that the act of conjugation of our barcodes to the antibody does not impair the underlying performance of that antibody. And also, in the way we design pairs of barcodes and label, we make sure that there's no cross-reactivity between any given two pairs. DNA amplification ties to the sensitivity of our platform, whereby we can elongate and extend the length of any given barcode to create a polymer on top of these barcodes, a bit like branches of a Christmas tree, on which you'll be able to attach many more labels themselves conjugated to fluorophores. As a result, you increase the ratio of barcodes per antibody, and therefore the amount of fluorophores that can hybridize through the label onto the antibody. What guarantees the reversibility of our platform is what we call DNA exchange, the act of dehybridizing at will the labels from their barcodes at any point in time. So how did we generate the 5-plex image that you saw before? After conjugating 5 different barcodes to the antibody specific to the markers of interest, we applied them in a single staining step. Subsequently, we apply a single amplification step to increase the relative ratio of barcodes per antibody. At this point in time, there's still no fluorescence on the sample. We come in with our first wave of labels that will hybridize to the first marker, in this case CD3. We switched on CD3, we acquire an image, we come in with our proprietary buffer that will reverse the hybridization, dehybridize the labels from its barcodes, and we can wash away the fluorescence from the sample. We're back to zero fluorescence. And we can iterate as many times as you want. The second step was switching on CD8 by introducing the label specific to the corresponding barcode. Using the same channel, we're using the same fluorophore in this workflow. After acquiring 5 different images of the 5 different markers, we can overlay them using a DAPI counterstain. As you can see, this method of sequential multiplexing is fully reversible. It relies on a single staining, single amplification step, and it starts without fluorescence and ends without fluorescence. To illustrate our DNA-mediated amplification method, we've compared the results that we are able to generate with our method on PD1 in tonsil tissue versus a traditional dilabel secondary based amplification method. As you can see, with our amplification method, we're able to generate a much higher signal to noise ratio. These images, left and right, were acquired on the same instrument, same exposure time. Our approach to remove the fluorescence from the sample by dehybridizing the labels from their underlying barcodes relies on a method that fully preserves the integrity and antigenicity of the sample. We avoid the use of microwave, heat, light, peroxide of any other detergent, and we don't change the pH nor the salt concentration of the buffer in presence of the sample. We use an enzyme whose function is solely to dehybridize the label without damaging the antigens and while preserving as well the barcodes and the antibodies that always stay on the sample. To demonstrate this full preservation, we've done multiple cycles looking at the same two markers, in this case cytokeratin and Ki67, over 6 consecutive cycles of hybridization and dehybridization. As you can see, the image that we obtain before and after these cycles were exactly comparable. The same experiment to demonstrate the full reversibility of our reagents was performed by our collaborator David Rimm at Yale Pathology Labs. He used the same markers cytokeratin and Ki67 over a number of consecutive imaging rounds and demonstrated likewise the full removal of fluorescence and the full comparability of the subsequent images he was able to image after re-hybridizing the labels on the same barcodes. Our collaborator David Rimm also assessed the intersample reproducibility of our in situplex platform by comparing two serial sections together looking at CD3 cytokeratin Ki67. He analyzed the results using a highly quantitative software platform, the Aqua platform, and observed exactly comparable results across the two sections. Thanks to the reversibility of our in situplex reagent platform, we can compare head-to-head images acquired in immunofluorescence with images acquired with traditional IHC. In this example, we first acquired CD3 in NSCLC using our immunofluorescent-based approach with labels attached to fluorophores. Then after removing the fluorescence from the sample, we came in with an HRP labeled secondary antibody that we applied on the same tissue section and generated the corresponding chromogenic image that we could in turn compare to the first immunofluorescent image we had generated. How can you access our technology today? We currently engage with customers through selected pilot studies, where we act as a CRO. Customers will select the markers of interest, we will design and develop the corresponding assay, collect samples from our customers, run them on their behalf, and return the raw images to them that the customer in turn interprets. A typical project flow lasts between 3 or 4 months, depending on the level of multiplexing and the complexity of the underlying markers. To conclude, I will leave you with a typical example of a pilot study, in this case a 7-plex acquired in NSCLC FFPE samples, looking at CD3, CD4, CD8, granzyme B, TTF1, PD1, and DAPI. The corresponding raw images were formatted to be analyzed on the Halo platform for further cell segmentation. I hope you've enjoyed this webinar. Feel free to visit our website and to reach out to us for any questions. Thank you.